just actually before jumping into the science, I can describe the Tri-Service Microbiome Consortium. So the journey to that was actually quite an adventure in coordination and collaboration, which has been a highlight at this meeting. It was definitely something that Colonel Hoisington pointed out because it's critical to accomplishing the mission in many spaces. And from the DoD perspective, we have a very unique mission space as well, which has also been mentioned. And that actually has led to multiple opportunities for collaborating between the different services to advance DoD research. Now, one of the things, as simple as it sounds, communicating and coordinating between different organizations, especially in the different services, is integral to our efforts. And so actually, starting back in November 2015, when meetings started occurring, we ultimately, the community, the DoD community, this was all the services identified that would be incredibly important to have a forum, a mechanism to permanently establish a group that would be the vanguard of microbiome research in the DoD. And so that ultimately, through other mechanisms at the Office of the Assistant Secretary for Defense and Research and Engineering level, so this DoD-wide policy-making groups, we received tremendous support for having permanency with this group. And back in December of this past year, so 2016, Dr. Patrick Mason, who was at that time director of the Biotechnologies for Health and Performance Council, which is also another DoD-wide organization that actually monitors ongoing biotechnology within the DoD that's of interest to not only our own research efforts, but those also that competing nations or organizations might be using. So keeping abreast of the current technologies that are being advanced. And Dr. Nancy Kelly Lochnane's here, also been a leading member of the BHPC. And a lot of members here actually also part of that group. So within that group, it was found that it would be extraordinarily valuable to have a Tri-Service Microbiome Consortium, a group focused on that DoD biotechnology policy direction, but focused on the microbiome. And so I'm very pleased to say it was on the 6th of December, Dr. Mason signed off on that charter. And now, as Dr. Mason actually has transitioned over to ONR for a new position, Dr. Ben Petro is taking the torch and leading forward. So all that ultimately has also resulting in us on the 10th through the 11th of May hosting a Tri-Service Microbiome Consortium meeting in the National Capital Region. It will be right outside Washington, D.C. So if you have questions about that, we'll be bringing in speakers from across the DoD enterprise to discuss on the quite broad array of microbiome interests, some of which we are hearing today. So to jump into the work that I'm going to present, we are one piece of that group, that team. And so I am currently looking at the U.S. Army Center for Environmental Health Research located on Fort Detrick, and generally that has enabled us, that focus has been in that organization on environmental health surveillance. So I'm going to tell you about one of the projects that we are advancing in our environmental health program that's leveraging the microbiome as a surveillance tool, so very much as Dr. Gilbert was discussing this forensic capability of looking at the microbiome. Next, I guess I have the command here, awesome. All right, just before, these are my opinions are my own and do not necessarily reflect those of the Army. So as I was mentioning, we are the U.S. Army Center for Environmental Health Research. For those of you that are not familiar with the medical research organizational structure of the Army, we actually fall under the Medical Research and Material Command, which is headquartered out of Frederick, Maryland, just 40 minutes outside of Washington, D.C. And our mission space has been to essentially the surveillance of environmental, physiological, and psychological exposure. So that alone is rather broad and comprehensive. So it has enabled us in a very unique way to investigate many issues. Historically, our focus has been on environmental challenges, things like environmental toxicants, things that are the warfighter downrange in the deployed environment would be encountering, things like burn pit exposures. And once again, topically addressing some of these problems, but there's other team members in the audience here, like Dr. Cam Mozzie and the team from the AFRL, who are also advancing this and with whom we collaborate on these studies as well. And so this mission is really, if I was to summarize, it really has been a center for systems biology integration. So taking all these complex problems that we encounter in the DoD operating environments and transforming the data or the problem into a systems biology oriented solution. So today I'm going to be focused particularly on the environmental toxicant aspect of our work, and that is a historical mission that the USACER the acronym for the U.S. Army Center for Environmental Health Research has been pursuing. And so to do that, I just want to give a brief introduction to this idea of the exposure effect continuum. So if you think from a toxicological standpoint, if you are exposed to some chemical or agent or compound or environmental stressor even in the environment, that ultimately in this black box idea of having some effect on the human body ultimately manifests in a clinical disease. So through this, paradigm that was set out by the National Research Council back in the 80s, you can actually understand this, you can better demarcate in the detail the processes that are occurring. So from that exposure, ultimately to the dose of that chemical that's absorbed into your body, going through the system to what's a particular organ might be experienced, and then the protein of interest. So if it's like a cytochrome P450 or some other protein of interest that's affected, that affecting that protein resulting in altered structure function. So like enzymatic inhibition, for example. Say if it was like an acetylcholinesterase, protein, and you have like a organophosphate pesticide which would inhibit that. And ultimately, because of that altered function, that's what ultimately transforms into clinical disease. And so one of the key approaches that we use at the USIS for all these different environmental stressors is looking at these biomarkers of exposure or susceptibility. Essentially, how early in this process, as this disease is evolving, how early in this process can we identify veritable markers that you, number one, have been exposed to something, which is extraordinarily important from the longitudinal health surveillance perspective, but also are there things that we can detect that might indicate that, wait a minute, you should not be in that environment because there are residual levels of this compound and we know that you have a signature that renders you susceptible. So if you look at this as a timeline, a temporal evolution of a disease, you can actually, if you think about it, and it's the reason why we're all here, when you add the microbiome, it actually represents the first interface that we can leverage these biomarkers of exposure. So the very, all of the surfaces of the human body and the internal linings all can host the bacterial, fungal, and viral components that are, part of the microbiome. So these signatures, having this ability, it is our first line of defense and first opportunity for surveillance, which is the motivator for our work. And so this is actually really important too, because not only from a surveillance perspective does it present a very attractive target for military health surveillance, but if we think about this too, in the staggering amount of literature that's now coming out on the ability of the microbiome to modulate chemical toxicity, but also, in a perfect example is antibiotics, the ability of chemicals to modulate the microbiome. And that's sort of, we, in a joint effort with the Army Public Health Center and the Dahlgren Naval Surface Warfare Center, drafted an internal white paper that actually explored, through a literature survey, the effects of the microbiome on toxicity. So the ability to take these environmental stressors and transform their toxicity from what their parent toxicity might be, and that ultimately having an effect on the host. At the same time, the host, through the toxicants being able to modulate the microbiome, once again, antibiotics are a perfect example, that ultimately is shifting the microbiome itself, and then this dynamic circle is cycling and potentially resulting in clinical disease of significance. So, and Colonel Hoisington definitely commented upon this, but the motivation from the military environment, this presents a very unique situation because we are asked to go into extreme environments and in situations that are very unique. And from a public health perspective, and Dr. Gilbert was, talking about this from a forensic capability, if you knew that there were unique signatures from being exposed to things like vaccines, from handling chemicals, from medical treatments, all these signatures on the microbiome, obviously, correlating to your own baseline microbiome, all these signatures could be used from a surveillance perspective for whatever purposes, be it for national security or for public health. And so, this is essentially, the figure highlighting these longitudinal exposures. If we are able to profile biobank specimens, be it fecal material or blood, if we can track that longitudinally from a pre-deployment to a deployment standpoint and for a military service member, one of the most important things, and we have Dr. Brenner here and from the Department of Veterans Affairs, knowing what you're exposed to when the service members retire and leave the service, and providing them with appropriate health treatment if they were exposed to something in these extreme environments. We need to improve the methods by which we can identify that people are exposed to a particular compound or situation. And so using the microbiome, that could be one of the solutions to that problem. And so, as much as we are a center for environmental health research, we're even more so a center for collaboration. And so, unfortunately, I'm not going to have time to go into this, one of the major aspects, which is actually using a notobiotic animal model. Dr. Chrissy mentioned that is one of our valuable models. But needless to say, at the UCC in our microbiome initiative, we've assembled a team of teams, and that's how you get this done. And that's been highlighted. That will continue to be highlighted. That bringing great people together gets the work done. So not only have we collaborated with our fellow Army agencies, our partners in the Air Force, but also reaching out to industry and academia to develop these capabilities resources. We have at the UCC our notobiotic animal facility, so getting that online was something we worked very closely with Open Biome, a nonprofit stool repository, and Charles River Laboratories, as well as working with other organizations in the team to develop unique projects, which unfortunately we don't have time to go into today, but I'm very, very happy to discuss offline. So today, I'm actually going to describe a really exciting study that we did. This was actually one of our first entries into the microbiome, and actually a collaboration with Dr. Rob Knight at the University of California, San Diego. And so just from this study, from a toxicological perspective, we actually had conventional Sprague-Dawley rats. These rats were exposed to 5 different chemicals, either sodium arsenite, cadmium chloride, sodium dichromate, cobalt chloride, nickel chloride. These are heavy metals. They're present in many things, all the way from consumer products to metal alloys to munitions. So these are militarily relevant compounds. And essentially there are 2 experiments that occurred. One was a 5-day exposure of the rats to these toxicants. And fecal material, in this case, is a relatively simple microbiome experiment, fecal material collected pre and post exposure. To identify any biomarkers of short-term exposure, we also had a single-day exposure. And from these samples, which the data actually were in, just preparing to submit a manuscript, we're able to take the fecal samples from the rats and then do 16S ribosomal RNA sequencing. That was done in collaboration with Dr. Knight's lab. And in addition to that, we also were very fortunate and able to get full metabolomic sequencing of these samples, so quantitative metabolomics and lipidomics to associate that with the deep sequencing metagenomic data, which in collaboration with the U.S. Army Medical Research Institute of Infectious Diseases, we were also able to develop. So this is, as it's being highlighted, these omics approaches to studying the problem, to have all these data, if you're fortunate and able to collect a sample that enables these studies, that's how you have a better picture of what might be happening if you're exposed to a metal while assessing the microbiome. So just briefly, just to go through some of the microbiome data, so everyone's probably familiar with these compositional plots, but this is at the very high order. So, and I'm just, for the sake of time, only showing one of the metals here. This is nickel chloride. And so what you're seeing essentially are the pre-exposure controls, the day 0s, and then the day 5s, which are the post-exposure. So essentially pre-post exposure to nickel chloride. From a phylum level, so you'll see a couple paradigms here. In the higher level orders, you can see disappearance and appearance of new groups of bacteria, or at least expanding into the environment that has been generated after the exposure to the metal. The great thing about 16S is that if you have depth of coverage, you can delve deeper into the data. And so then you can start, from these, plots, you can start defining the components and the constituents of these different bacterial groups down the order classification. Ultimately, in these graphs showing at the low dose, we had multiple doses in these experiments, but I'm just showing this as an exemplar here for the low dose. Ultimately, what you can see is not only, in the appearance of, and especially in the verruca microbia, which are the purple groups, disappearance of entire groups in the post-exposure, so an environment being created that would enable these other bacteria to fill that niche. But also, you're seeing also a groups such down here, such as in the Bacteroides, where the proportion of the actual bacteria is remaining relatively similar, but some of the constituents at the family level are changing, and this is very evident in this group, this red group that disappears, which is S24-7, which some of you might have encountered in your own studies, which is a notorious member of that's being characterized better, but one of the components that we saw in these studies. So not only from just exposure to the metal is this important, but from a dosage. So these were different dosages selected just based on some in vitro data as well as historic literature mining and effects we saw. And what you can actually see is in a dose-dependent, if you go back and forth, you can see in a dose-dependent manner, you can, the composition of these bacteria, you can accentuate the developing signature. And so this is incredibly important because the objective of this original study was from the environmental surveillance and to identify changes in the microbiome that we could use as signatures of exposure. So just relatively quickly, so this is sort of a highlight, an aggregation of all the data of all the metals, and what you're essentially looking at is in each one of these columns, you can see the different toxicants that we used and the three different dosage levels. So that's evident in the colors at the top. And what you're looking at are essentially wald tests of the pre-exposure versus the day 5 exposed samples. And so what you're seeing is essentially a heat map of bacteria that in a log-fold change either were increased in the post-exposure or decreased. And so what you can see, just without getting into the details, is that there are patterns emerging, especially for the different metals, groups of bacteria, interesting ones, and not time to delve into any of the specifics, but getting these signatures of exposure from the different toxicants at the different doses, and that enables us to start creating a model of what might be happening. And so sort of just to wrap up right here, just as a summary, what you can do is then essentially just take all the significantly changing bacteria, and so essentially from a genus level, if you take the most significantly changing groups and actually plot them, and with respect to the toxicant that the animals are exposed to, you can now start getting those signatures, using the rat model that we use, signatures of exposure. And so this is very important because not only this is from surveillance, but when we discuss the warfighter performance, are these bacteria that are changing, are they ones that are disappearing and we need to supplement them in some way if we were to go the direction of probiotics? Or if they are ones that are appearing, are those ones that we need to account for and try to eliminate that might be conferring a deleterious effect? So overall, it's exciting work. This was with conventional rats. We've now expanded this work to humanized mice, to conventional mice. So a lot of great work and with some fantastic partnerships. And probably the most important slide here is that these are the people that are getting this done. It takes a team of teams, fantastic collaborations across all the organizations, not only in the military, but also with our partners outside of the DoD. So thank you.